Oh, so MJ is controlled by the button?
Yay!
Should be okay.
Well, can we just
That should
turn
be
down
okay.
these lights a little bit?
This looks good. I can see everything.
Wait, it's not a dimmer? Oh, hey, oh, yeah, it's
Button 2.
Okay. Oh, that's
There you
good.
go. That's nice.
That's
Can
good.
you stop it
I
there?
can't like stop it.
That's perfect.
Now we
That's
can't
perfect.
see.
That's perfect.
Nice technology.
Can you still see? Can you still
No,
see her?
I
though?
can't see her.
Yeah. Very... Why can't I can see
No, you got it.
Can't see her at all.
Is there a way to? Is there a light here? Or like, what about these lights? There's some.
Oh, too bad.
No.
Well, I'm so glad you're here.
Oh, that's totally
You got some time though, right? Dwayne,
This is
my cousin
no, I think those are the same kind of
Suzanne.
Yeah. Is it on the second
Come all the way.
Yes.
And this is my
I think
sister.
it's the same.
There must be something. All my friends. Wayne, friend of mine also.
It's like totally dark.
Can you put a? Is there a light on the projector? No, we tried that.
Fiddling people. I made sure that. Oh
It's, you can see just the face. I can actually see Patrick more clearly than I can see you, Maria. I can see.
Wait, how is this?
It's okay. Still not great.
What about this?
I think the thing is, you're wearing a lighter shirt, and that's why it's showing up, as opposed to Maria.
Isn't there a way to put more lights back there? Oh, that's too dark. Oh wait, so Maria, this is mom.
Hi, Maria. Oh my God. Yes, Maria.
And this is MJ. And Zach, MJ. This is my big brother.
What you get?
My son here.
I left last Thursday.
Oh, yeah. There's everything on the sun there.
Oh, no, sweetheart.
Alright, I think the best option is to keep some light on there.
Good luck, Padmini! Dr.
Hey, Bono!
Hi, Bonos. I love you.
Good luck. You're going to be Dr. Bono today.
Who's watching?
Well, everyone, yeah, currently.
Oh, who's watching and has a webcam?
Dr. Bruno!
Yeah, so it should be searchable on YouTube, hopefully,
Okay.
for the next 8 hours or so.
Do you know if you happen to send it to my mom?
Rango something?
That's my dad. And then
okay.
the Revati.
I had it, yeah. I
Okay,
think.
great.
There's no,
Excellent.
let's see. Oh, shoot, I should have sent it to myself to confirm, which is, in hindsight,
Well, I assume if they got it, then.
yeah, can you text them and just check in case? So I sent it to these, that's her, right?
Yeah, let's open just in case we can see.
Let's just see the file.
If I click on it, like, can I see what the actual address is?
Apparently not.
Well, but if you didn't get a bounce back.
I didn't get a bounce back.
Okay,
But if
I'll check.
your dad put together.
Yeah.
Okay, so at least one of them.
Yeah, hopefully. Yeah.
Yeah, but just like, let me know.
Will do. Will do. Thank you. Okay. Okay.
So they may be other buttons but they should, only one should be live streamed.
Okay.
So they should, and I put that in the email. So they should be able to see, but let me know if they can't.
Will do. So I'll call them in a second.
Hey,
Oh,
hi.
nice to see you again.
How are you?
You guys hear me now? I'm not.
Nobody's else to figure it out. What's her seating figure
I'm gonna see here so we can. Dude, you're so tall.
I can hear you in the hall. She's been saying that for years. You guys went up there.
Yeah.
And then I was like,
I'm gonna get a picture right now. Otherwise, it was not gonna work.
Oh, yeah, what's that? That's what.
Yes. That's sweet. Go.
What?
Is it too late? Can I add a couple of people?
Congratulations, dude. Okay, well, congratulations in the future.
But you can just tell them to search for you on YouTube.
Okay. You can tell, maybe if you tell my auntie Suzanne, she can like email the family or something.
Do you have, yeah, so you could just type in.
Can you text me that watching?
Oh, wait, you can see me?
I'm just saying.
Okay.
Okay,
You
great.
know what I'm saying. Family.
You can see
You can
Padmini.
just text it to me.
Who's that?
Is that Hannah? Hello.
Yeah.
Who's that guy?
No, they can see you.
It's James.
Oh,
I don't know.
Oh, it is? Oh yeah, I see that you're on, Alma. I think you just have to turn your webcam. No, she's able to see it. She was like, how come you can't see it? Yeah, I can see you, Alma. Wait, Amma, your webcam is where your computer is.
Was it your mom?
Look at the look at the computer. Look at the computer, cause yeah, exactly. Oh, now she's gone.
Why? Because she has it on the TV?
Yeah, they're projecting it on the TV.
That's funny.
All right, good. Thank you.
I think she wants to send that to 3 more people, but I sent it to her email, so I actually don't have access to it. So is there any way you could I don't know who she wants to send it to, so I don't know.
Yeah, let me ask her.
I forgot it.
I think I'll take like 1 year off. I think I'll take
Thank you.
Yeah, she did. The only thing I'm thinking is in case this runs out,
Right.
back up, and mine is too short to go from there to there. So, do you know what I mean?
I think she did bring her long one.
Okay. Well, it should be okay. It's at 100%. So as long as they don't go over an hour.
And your battery
It's
is
85%.
notoriously unreliable.
Like,
I would feel
enough.
safer.
It's 85%. It should be okay.
Yeah, it should be.
All right. Okay. I want her to have any case.
Yeah, exactly. I think she charged it full weight. Where are you sitting? Can I sit over here? That's what you get with the company. It could just be that there's something. Yeah, if you just go it should.
So Padmini started to work on a project that sort of challenges the current problem. And so for over 10 years or so, researching immunology, despite a lot of effort by many, including ourselves, hasn't been able to reveal the key innate sensors that detect the virus and that defends the host against influenza. And so Padmini wasn't quite happy with this result. So she challenged this whole field by creating mouse strains that actually is appropriate for this position. So I don't want to steal any further from her talk today, but essentially what I want to say is that Padmini is a fighter. She finds problems, she tackles them with fierceness, you know, she's kind of, and she is determined. And one of the things that really strikes me about Padmini is that she never is afraid to do anything. So usually we have a one-on-one meeting in my office and we discuss something and it's completely outside her field, comfort zone, whatever. She'll say, okay, I'll do it. And next time we meet, she has beautiful data, you know, but she's not only just doing things herself, she's able to recruit experts from within and from outside to get things done. And that's one of the sort of key features that makes her so tough. She's able to recruit experts from within and from outside to get things done. And that's one of the sort of key features that makes her so tough. She's able to recruit experts So,
Quick one. Okay. Hi everyone. Welcome to my dissertation defense. So today I'm going to talk to you about my project, as I said, on influenza virus. And so my thesis focuses on how flu infection is recognized by the immune system and how this recognition can lead to either antiviral responses and protection or lethality in disease. And then we'll talk about potential strategies to improve survival after flu. Okay, so you probably had flu at some point. Annual flu epidemics cause 3 to 5 million cases of severe illness each year, resulting in around 500,000 deaths. And this image here is from a satirical French magazine from the 1800s, illustrating that chaotic response that ensued during epidemics. Still kind of occurs. And influenza imposes a considerable economic burden all over the world. In the United States alone, recent estimates put the cost of influenza epidemics to the economy at around $100 billion per year. And in 1918, the infamous flu pandemic, also called the Spanish flu pandemic, infected over 500 million people. Estimates showed that 3 to 5% of the entire global population died. And the flu virus is constantly mutating, so small mutations happen continually, but large genetic changes also occur that enable the flu strains to jump from species to species. So the threat of a swine flu or avian flu pandemic is always possible. Another challenge regarding flu infection is depicted in this graphic. So you can see that with each decade after the age of 55, death from flu increases remarkably. This amounts to 90% of flu-related deaths occurring in the elderly. But the underlying mechanism remains unclear. And although the virus itself can cause significant damage to respiratory tissues, secondary bacterial pneumonia is the common cause of death in people infected with seasonal flu. And analyses of the host response to both pandemic flu strains or flu infection in the elderly show how it's not necessarily the virus itself causing death, but the excessive post-inflammatory response to the pathogen causing the problem. And another obstacle we face is that the current flu vaccine does not provide long-lasting cross-protective immunity. So what exactly happens when flu infects a host? Well, this diagram over here, he depicts a susceptible host. So a host becomes susceptible if they are unable to reduce the amount of pathogen or unable to tolerate the negative consequences of the host's immune response to infection. So this frequently happens when the elderly are infected with flu. There are two distinct host strategies to respond to an infection: resistance and tolerance. So a resistant host regains health by recognizing and eliminating the pathogen. And we currently have therapeutic strategies that attempt to boost resistance to flu. So for example, the annual flu vaccine tries to boost resistance by stimulating the immune system so that it can more easily recognize and destroy any flu the host may encounter. But as I mentioned, flu vaccine is not always protective. There are also antiviral drugs like Tamiflu that disrupt the ability of the virus to spread from cell to cell. But these therapies don't work very well because by the time you're diagnosed with flu and you're prescribed it, the virus has probably infected you quite well. Another strategy a host may use to respond to infection is tolerance. So a tolerant host regains health by reducing tissue damage that's inflicted by either the pathogen or the host. So like the susceptible host, there may be large amounts of virus present, but unlike the susceptible host, the tolerant host doesn't overreact to infection or cause excessive inflammation. The tolerant host has a chance to repair any tissue damage. So boosting tolerance to flu could greatly improve the outcome of flu infection in the elderly. But currently nothing is known about how to boost tolerance to flu infection. So there are currently no drugs that take advantage of this strategy. So to outline my talk for today, I'm going to give you a little more background about my project and the rationale for asking the questions that we did. And then I'll tell you about how the immune system triggers antiviral resistance against flu infection. Then I'll tell you about how recognition of the virus can also lead to lethality. I'll also discuss potential strategies to prevent lethality and boost tolerance to flu infection. And last, I'll discuss the conclusions and implications of my data. So let's dive into the background. So influenza virus, or IAV, is a negative-strand single-stranded RNA virus. So in the core of the virus, that single-stranded RNA wrapped around nucleoproteins called viral RNA. And it has polymerases that are attached to it that help it replicate in the host cell. And on the surface, the virus expresses hemagglutinin, or HA. And this is what the virus uses to attach to the host cell and initiate the process of entry. And neuraminidase, or NA, is essential for new viruses in the cell to be released from the cell surface. And so influenza subtypes are named based on these two proteins, like H1N1 or H7N9. Influenza also expresses this M2 proton channel on its surface. So this protein helps release viral RNA upon entry into the host cell, and once new proteins are made inside, the M2 prevents premature fusion of new viruses in the cell. So during flu infection, the virus binds to the target cell using HA, and HA binds to sialic acids on the surface like glycoproteins. Then the virus enters the cell by a process called endocytosis. And while the virus is in this acidic endosome, that flu M2 protein acidifies the virus core so that the single-stranded RNA leaves. And then it travels to the nucleus where a messenger RNA is made to make new viral proteins. And new viral RNA is made called 5'5-phosphate RNA. And so this new RNA, this 5'5-phosphate RNA, will go into new viruses that are being made. And so once it exits the cytoplasm, then the new viruses are assembled and they bud from the cell where they can go on to infect the next cell. So as you can see, there are many steps to this flu replication here that can be detected by the host. So how does recognition of flu occur? How does the host cell recognize what's happening here to mount an immune response? Dr. Charles Janeway, who started our immunology department and is considered the father of innate immunity, provided a conceptual framework for the current understanding of innate immune recognition. So back in 1989, as an introduction to the Cold Spring Harbor Symposium on immune recognition, he stated that the most likely possibility is that primitive effector cells bear receptors that allow recognition of certain pathogen-associated molecular patterns that are not found in the host. I term these receptors pattern recognition receptors. So the innate immune system evolved to detect the presence of conserved pathogen-associated molecular patterns in plants using pattern recognition receptors, or PRRs, to trigger host responses that limit pathogen behavior. So there are two arms of the immune system, innate and adaptive immunity. So the innate immune system is ancient. It's been around since Metazoans began to form around 600 million years ago. It's found in plants and fungi, insects, and primitive multicellular organisms. So upon recognition of plants by PRRs, innate immunity is rapidly initiated by sentinel cells. So for example, epithelial cells act as physical barriers to pathogens, and innate cells also recruit other immune cells to the site of infection to clear invading pathogens. Innate immunity also evolved to activate the adaptive immune system. So, by contrast, adaptive immunity is young. It's only 400 million years old, approximately. And so, adaptive immunity depends upon diversification of antigen receptor genes to generate this vast repertoire of lymphocytes, each which expresses a different antigen receptor. So basically, after infection, innate cells of the innate immune system can digest the pathogen at the site of infection, and then they display small fragments of the pathogen called antigens on their surface. And these antigens present themselves, then travel to the lymph node or the spleen where the T cells are, and where those cells, the adaptive immune system can recognize their antigen, and then they can proliferate and go back to the site of infection, kill infected cells and neutralize pathogens. And a small number of those T and B cells survive for decades as your memory pool. So upon reinfection, those memory T cells and B cells can quickly eliminate the pathogen again. So that's the whole concept behind vaccination. But for innate immunity, it's pretty much the first time every time. So my project mainly focuses on this frontline effects that uses pattern recognition receptors, or PRRs, to sense infection, to eliminate any invaders, and then also activate adaptive immunity. So to develop effective therapeutic strategies against the flu, we need an in-depth understanding of both innate immunity and adaptive immunity to the virus. But how do PRRs of the innate immune system recognize flu infection? Well, when influenza enters the cell through the endosome, a sensor called TLR7 recognizes the single-stranded RNA of the virus. And after the virus replicates in the nucleus and those new 5' triphosphate RNAs are made, they are recognized by a sensor called RIG-I in the cytoplasm. And activity of new viral proteins, particularly that M2 protein of flu, is recognized by a complex called the inflammasome. So the inflammasome can be activated by cellular stress, damage, bacterial and viral infection. So the influenza M2 activity is detected by the inflammasome and activates the enzyme Passage 1. So let's talk about what these PRRs do. So TLR7 and RIG-I, using that adaptive mass, results in the production of crucial antiviral proteins called type 1 interferons, or interferon alpha and beta. So when TLR7 or RIG-I trigger the production of type 1 interferons, it binds to its receptor, if not, and this leads to transcription of hundreds of interferon-stimulated genes, or ISGs, including my favorite, MX1, which we will talk about very soon. So the products of these ISGs, these genes, are proteins that exert numerous antiviral effector functions. So basically, type 1 interferon, downstream of the PRRs, TLR7 and RIG-I, lead to antiviral responses. In addition to interferons, TLR7 and RIG-I, via that adaptive mass, also trigger the production of pro-inflammatory cytokines IL-6 and TNF-alpha, and the proforms or inactive forms of the cytokines IL-1 beta and IL-15. And so when the inflammasome gets activated and Passage 1 gets activated, this results in the cleavage of those proforms into their active forms, IL-1 beta and IL-15. These cytokines are important for recruitment of immune cells to the site of infection. So this might look pretty redundant, right? But these pattern recognition receptors recognize different structures or different camps of flu, as you can see. And they're expressed in different cell types and in different parts of the cell. For example, TLR7 is in the endosome, and RIG-I in the inflammasome, or in the cytoplasm, doing different things. So to study the role of these pattern recognition receptors during flu infection, in-bred lab mice have been created that have genetic deletion of these different PRRs and signaling pathways. So a very common lab strain is C57 black 6 mice, or B6 mice. And so B6 mice lacking TLR7 or NAS or Passage 1 and 11 have been bred. And so after flu infection, TLR7 and NAS knockouts have no change in body weight, viral titer, or survival. And studies from our labs and others demonstrated that in the absence of components of that inflammasome or receptor, or in the absence of Passage 1 and 11, that mice lose weight. But you can see here that they only have an increase in viral titer around day 7, and they die around day 11 because they can't trigger adaptive immunity. So you can see there's a decrease in CD8, CD4 T cells, and there's less antibodies. But this calls into question, what are the goals of these PRRs in innate antiviral immunity? What's going on during the first 5 days of infection? So studies in these mice have not revealed any dependence on PRRs for innate resistance. And understanding innate immunity to flu is critical to our understanding of what's happening in humans during flu infection. And we know that both TLR7 and RIG-I recognize viral RNA. And in fact, the flu expresses an NS1 protein, and this NS1 protein is evolved to block RIG-I signaling to evade the host and prevent interferon infection. So clearly RIG-I and TLR7 are important, but what's going on? Why don't we see it here? Well, it turns out that these inbred mice This paper is from 1961. Shortly after discovering interferon, John Lindemann observed that a particular strain of mice called A2G mice were completely resistant to intracerebral and intrathecal inoculation of flu. So in this figure taken from this paper in virology, the inbred mice, that you can see here, all of these, died at basically both doses of flu infection, while those A2G mice all survived basically every dose. So these mice were extremely resistant to infection. And years later, they discovered that this protection was due to mucivirus resistant protein, or NX1. And so this was the first interferon-stimulated gene that was discovered, and it's highly conserved. Humans express NXA, the same thing, which restricts influenza and a number of other viruses. And this protein is a diamine-like GTPase, and it basically forms a polymer or a complex around the viral RNA, so around the gene. And when GTP binds, NX1 twists and breaks the flu so that it cannot replicate in the nucleus, and primary transcription cannot occur. All right. So, what's the deal? The deal is that laboratory strains can't express NX1. Laboratory strains carry deletions or nonsense mutations in the NX1 gene. So, Balti mice and B6 mice have deletion in a coding region, resulting in premature termination of the NX1 protein. But what we have done is we've obtained NX1 conjugate mice on the B6 pattern. Basically, these mice are B6 mice that can now express NX1. Like wild mice, they have the NX1 gene replaced with promoters. And so to compare their responses to flu, the LD50 of B6 mice, right, the guy on the left, the LD50 of B6 mice, so the dose required to kill 50% of the infected mice, is only around 25 PFU, while the LD50 of mice that can actually express NX1 again is nearly 7 million PFU. That's a lot of food. So our hypothesis was that innate and adaptive immune induction in mice that can actually express NX1 must be dramatically different. And this is indeed so. So in this experiment, I infected B6 mice and NX1 mice with different doses of flu. So PFU means platform units, or it's a measure of the number of infectious virus particles of flu. So you can see here, in these after I give those inbred mice that don't have NX1, 1,000 PFU, they lose weight very quickly and die on day 6. In infection in NX1 mice, after 10 PFU or even after 1 million PFU, I needed 5 log orders more than this guy, 5 log orders more to eventually see weight loss, and they died on day 3. So they're extremely resistant. And so on the bottom here, I looked at viral titer. So I infected B6 mice and NX1 mice with flu and measured viral titer on day 7. And NX1 mice infected with 1 million PFU are already clearing the virus, whereas these guys who only give 10 or 100 PFU can't control the virus at all. So the question is here, okay, so these NX1 mice are extremely resistant. But is it because NX1 is resistant to flu, or is it because they've triggered adaptive immunity? Just to double check that, we measured both T cell and antibody responses after viral challenge. And so after infection in these B6 mice with 10 or 100 PFU, robust induction of flu-specific T cells. After I gave NX1 mice 10 PFU, I couldn't even generate much of a CD8 T cell response, presumably due to robust immune control of virus infection. But here, after 1 million PFU, we finally see that and these guys died before they. But the point is, it's probably not due to innate immunity. And here, you're looking at antibody titers. These guys generate basically the same amount of antibody. If anything, the NX1 mice generate less antibody. So it's not due to induction of adaptive immunity. It's because there's just robust innate control due to NX1 expression. So based on this data, it was likely. So based on these data, it was likely that after innate recognition of the virus by PRRs, mechanisms of resistance and tolerance in NX1 mice could be very different, more closely mimic what's happening in humans after flu infection. And since NX1 null inbred mice have been unsuccessful at revealing the mechanisms of immune resistance, my project uses NX1 mice as a tool to revisit all of this and ask the question, how do pattern recognition receptors play a role in protective immunity during flu infection? Now, obviously, mice and humans are very different, but now we have a slightly better genetic background on which to ask this question. So for the rest of the talk, all the mouse data I'll show you is in mice that can actually express NX1. Okay, so the first thing we did was we took our mice and we made NX1 mice that were lacking these PRRs to investigate their role in innate immunity to flu. So here I challenged mice with 1 million PFU of this PRA strain. And you can see the genetic deletion of caspase-111 in blue or TLR7 in green did not result in any weight loss or change in survival. But unlike inbred mice, now when mice lack NACs, which is that adapter for Riga, they start to lose weight and they succumb to infection on day 7. Mice lacking both TLR7 and NACs in orange die a tiny bit faster. And here, this absence of MAPs correlates with an inability to control virus. So this is just looking at viral titer in the bronchoalveolar fluid. You can see these guys have lower viral titer, but in the absence of MAPs, these mice cannot control the virus. So after high-dose infection in NX1 chondritic mice, single deletion of TLR7 or caspase-111 results in no change in body weight or viral titer. So, but unlike inbred mice, now these NAS knockouts, these guys that can't signal downstream downward eye on the NX1 background, have increased weight loss, viral titer, and mortality after high-dose infection. So we begin to reveal dependence on these PRRs for innate. But what about the other two pathways? Well, after infection with a much lower dose of only 100 PFU, now caspase-111 knockouts, TLR7 knockouts, and now even NABs knockouts can recover from infection. The only guys that die are the mice lacking both TLR7 and NABs. And they succumb to infection somewhere around day 10. And to probe the mechanism of protection conferred by NABs and TLR7, we examined type 1 interferon secretion in bowel fluid 2 days after infection. And you can see that interferon beta was greatly diminished in NABs knockouts. It was basically undetectable in TLR7 knockouts. So these double knockouts reveal a compensatory role of TLR7 in providing innate defense. And since we saw this difference in interferon, we analyzed the expression of NX1 in tissue sections. So here, all this intense red staining is NX1, and DAPI is just a nucleotide form with cell via those two images. So you can see that in the absence of caspase-111 or TLR7, this didn't really affect NX1 expression. Losing caspase-111 or TLR7 did not affect NX1. But mice deficient in NABs, the adapter or Riga, lost the ability to accumulate NX1 protein in airway epithelial cells. But there was still a low level of expression of NX1 in the parenchymal cells, and this was confirmed by FACS. So by FACS, I can see that TLR7 provided enough interferon to induce NX1 in parenchymal cells like PDCs. And lastly, if you knock out TLR7 in NABs, you lose completely. You lose NX1 expression completely, and therefore these mice are very susceptible to flu. So, Rigai and TLR7 confer antiviral resistance to the production of type 1 interferons in different cell types, and interferon can then induce NX1, which can then potently control flu relief. So next we sought to determine the determine the cause of death in TLR7 NABs double knockouts. So, in the absence of TLR7 and NABs, was it just because they couldn't control viral replication, or was it due to tissue damage? And also, what are the consequences of deficiencies of all three sensory pathways after flu infection? Okay, so in this top panel, I show you again that the double knockouts in orange succumb to infection and die on day 8. But additional deletion of caspase-111 in purple here led to less weight loss, and these mice actually survive. So basically, these mice die, and then if you go ahead and delete caspase-111 as well, now they survive. Interestingly, although these mice survived, they had similar viral loads compared to the So in the absence of TLR7 and MAPS, deletion of pathway 111 actually rescued the mice. Next, we looked at histology sections of lung tissue from these mice. So here you can see wild type and single knockouts had moderate inflammation around the airways and veins. But these double knockout mice that lack both TLR7 and MAPS had severe inflammation. And if you take a closer look here, they had severe inflammation around airways, leukocytic infiltrates including neutrophils, and they suffered from airway degeneration, hemorrhage, and epithelial and ventricular necrosis. But in contrast, those mice additionally lacking caspase 111 had much less severe inflammation. So to summarize, mice lacking TLR7 and MAPS have increased. Mice lacking TLR7 and MAPS have increased viral titer, weight loss, pathology, and mortality, mimicking a susceptible host. When mice additionally lack caspase 111, there's still high viral titer, but only moderate weight loss and moderate pathology, and these mice actually survive. So using MX1 mice as a tool, we revealed tolerance. We revealed tolerance phenotype. And these data can be relevant to human flu infections, particularly in the elderly that succumb to infection, because of data from Ryan, a talented grad student in Europe. So basically, Ryan he took monocytes from old and young donors and infected them with flu, and then measured inflammatory cytokines and interferon. So here, in these two graphs, you can see that IL-6 and IL-1 beta levels are the same between young and old donors. But there's much less interferon in monocytes from old donors. So here, flu-infected monocytes from older humans in red were able to produce pro-inflammatory cytokines, but there was significantly less interferon in older monocytes. So if you look at this heat map on the right, Ryan looked at gene expression from infected monocytes from young donors and older donors on the bottom. So red means high expression and blue means low expression. And each column is a different ISG, so a different interferon stimulated gene is blue. And you can see there's a lot more red on top in the young donors and a lot more blue on the bottom. So basically what's going on here is that old monocytes show consistently lower expression of ISGs known to have antiviral activities against flu, including MXA, that human ortholog MS1. Furthermore, expression of IRF7, which is a critical transcription factor for type 1 interferons, and STAT1, an interferon signaling molecule, is lower in the older cohort. Now, if you look all the way on the right, these two columns are looking at influenza gene expression. So now we see a lot more red on the bottom and a lot more blue on top because these older donors don't have interferon stop virus. So there's a lot more viral gene expression in the old donors due to a lack of viral restriction. And Ryan showed the same response in monocytes using synthetic ligands of Brigide, and we also have the same data in human macrophages from old and young donors. So, since interferon is triggered by TLR7 and Brigide via MAPS, we can use our flu model in MX1 mice to investigate possible in vivo consequences of weak interferon responses to flu infection, and the mechanism by which immunopathology, specifically downstream of caspase 111, can contribute to that. Also, we can now look at caspase tolerance. So, to begin to look at how caspase 111 could trigger damage and death, we thought about what happens downstream of caspase 111. So here, caspase 111 triggers the production of the active forms of IL-1 beta and IL-18. And when they bind to their receptors, they trigger downstream signaling that leads to immune cell recruitment. And both these receptors use an adapter called MyD88. So if we wanted to see what the role of these pathways were, what we did was we made mice that are lacking MyD88 and MAPS. And so you can see in black here that even though we knocked out MyD88 and MAPS, these mice still succumb to infection. And to further probe the role of IL-1, we use this drug called anakinra. So anakinra is an IL-1 receptor antagonist that's used to treat chronic inflammatory diseases like arthritis. And here you can see in brown that even though we treated mice with anakinra, they still succumb to infection. So basically we concluded that IL-1 and IL-18 aren't totally responsible for the death of the mass double knockout mice, since they still die on the clock. Okay, what else does caspase 111 do? One of the hallmarks of caspase 111 activation is the recruitment. So we sought to determine if neutrophils mediate disease and lethality of double knockout mice by completing neutrophils. And so you can see in blue in the middle that double knockouts that have neutrophils depleted lost less weight and they had improved survival. So these data indicate that even in the absence of innate resistance, even though the concept is still present, tolerance to flu can be induced by depletion of neutrophils. But what's weird is that when we looked at neutrophil infiltration into the lungs of double knockouts and those tolerant mice that survived, by both facts and immunohistochemistry, they had similar numbers of neutrophils. So we hypothesized that it wasn't actually a consequence of how many neutrophils were coming into the tissue. It was actually what those neutrophils were doing in double knockouts, their function that was detrimental. And one of the many functions of neutrophils is to mediate clearance of pathogens by extrusion of their DNA through mitosis. So once neutrophils recognize the pathogen, they decondense their chromatin by citrullination of histones, and then the chromatin is released lined with granular components like elastase and MPO and contestants. And this creates this fibrous net with antimicrobial properties. But these nets can also cause a lot of collateral damage. They have direct cytotoxic effects on endothelial cells, and which is consistent with that granular necrosis that we saw in double knockouts. Also, the histones in those nets can trigger caspase 111 activation. So this paper showed that when neutrophils are recruited to the lung, that the neutrophils perform mitosis, and those histones can go ahead and activate the inflammasome in caspase 111. And caspase 111 will trigger more influx of neutrophils, it triggers more mitosis, and perhaps a vicious cycle of inflammation happening. So our hypothesis was that influenza infection in double knockout mice was triggering mitosis downstream of caspase 111. And so to investigate this, we took sections of influenza-infected lungs and stained them for H3-CIT. So H3-CIT is a marker for mitosis, and that blue again is a nuclear stain showing us the abundance of cellular infiltration. So in these double knockouts, there's a lot of blue, and there's also a lot of H3-CIT staining in red. And this is almost absent in the lungs of the tolerant mice. So deposition of nets occurs downstream of caspase 111 activation and correlates with excessive lung. But now we have to step back and look at the bigger picture, because most of the data published about mitosis is related to bacterial infection, and in humans, the elderly are most vulnerable to the flu, but they mainly succumb to pneumonia caused by secondary bacterial infections. And a number of published studies show that influenza and consequent airway epithelial damage is sufficient to trigger bacterial colonization of the lungs and bacteria-associated pneumonia. Basically, when host defenses are weakened, nasal commensals and other bacteria in your nasal passage can migrate down the respiratory tract and expand in new niches in your lung where they can become harmful to the host. So we hypothesized that in double knockouts, perhaps bacteria, in addition to flu, is triggering the activation of caspase 111, which is then triggering mitosis and immunopathology. And then further deletion of caspase 1 leads to tolerance to both flu and bacteria. And we have some evidence of this, because when I inoculated those wild-type MX1 mice with the bacterial component LPS intranasally, I saw extensive mitosis in the lung, all that was staying there. This was absent in caspase 111 knockouts, indicating that the requirement for caspase 111 for net release extends beyond flu infection. So to test our hypothesis that in the absence of innate resistance, that flu infection was also triggering secondary bacterial infection, we infected mice with flu and just measured bacterial burden on day 7. So here you can see that both TLR7 and NABS knockouts in orange, and those tolerant mice in purple, had bacterial bloom in the nasal cavity. And we measured this by bacterial culture and PCR. And with the help of Angel and Chlorella, we observed that the composition of different bacterial genomes altered in those infected mice. Specifically, double knockouts and those tolerant mice had an increase in the abundance of members of the Pasteurella family. It's mostly live as commensals in the upper respiratory tract. But okay, so what? We see this in the nose, what about the lung? Well, analyses of the lung from those flu-infected double knockouts revealed a similar increase in bacteria, specifically from the Pasteurella This is confirmed also by bacterial lung disease mice. So these data indicated that bacterial outgrowth accompanies flu expansion in the respiratory tract of infected mice. So a failure to induce interferon promotes viral growth and tissue damage within the respiratory environment that is conducive to bacterial bloom of commensals. And this is very similar to what happens to humans during flu infection. So since humans are treated with antibiotics, and to examine the role of bacteria in driving lethal disease in our model, I infected those double knockouts with flu and treated a group of them with antibiotics orally starting on day 3. And here you can see that this improved survival. But since we're getting rid of pathogens, this is basically like improving resistance. But what about our idea of boosting tolerance to flu infection? So since our data suggests that any benefit that neutrophils might provide an antibacterial defense is likely outweighed by the damage they inflict on host tissues, the next logical step would be to inhibit the deposition of nets and inhibit caspase 111 signaling to see if we can induce tolerance in these mice. And the first way we try to do this is using palmizyme. So palmizyme is recombinant DNase, and it's given to cystic fibrosis patients because they have so much mucus in their lung, and so this palmizyme is given using a nebulizer, and it can break up all the DNA in the lung, loosen up that mucus so that it can be cleared so the babies can actually breathe. And so what I did was to see if net digestion can improve survival in the double knockout mice, is I gave palmizyme intraperitoneally throughout the course of infection. So I couldn't give it intranasally because that would just wash down with more bacteria into the lung and give them more bacteria. But even just giving it intraperitoneally improved survival. You can see here that the mice that survive actually return to their original weight in gray. So in the absence of innate restriction of both virus and bacteria, degradation of nets dampens that host-mediated inflammation and improves disease outcome after flu infection. So this is one potential way to boost tolerance to flu. So to summarize our data in MS1 mice, so using MS1 mice as a tool, we've been able to investigate mechanisms of resistance and tolerance. And then I'm showing you susceptibility as well. So let's start with a resistant host. So in a resistant host, after flu is recognized, type 1 interferon triggers the expression of MX1, and MX1 blocks viral replication, reducing viral burden. And studies also show that type 1 interferon can inhibit activation of the inflammasome and caspase 111 via SAT1. Okay, let's go back to the natural flora. So in a susceptible host, there is less activation of type 1 interferon in MX1, and so the virus is able to replicate. And that replication itself can cause tissue damage and facilitate bacterial infiltration. Flu also activates the inflammasome and caspase 111, which can lead to cytokine production and tissue damage. And now bacteria can further expand the lung. And this is further exacerbated by neutrophils in the tissue, releasing nets in an attempt to clear bacteria, but instead causing more tissue damage. And both nets and bacteria can further cause caspase 111 activation, leading to the cycle of the nephrology that I described earlier. Using MX1 mice, our data shows that we can use tolerance. So in the tolerant host, there's still this high viral titer because of the lack of interferon and the lack of MX1. And that high viral titer can still trigger tissue damage and bacterial colonization of the lung, but there isn't that propagation of that caspase 1 and neutrophil vicious cycle. So older adults suffering from flu infection might benefit from therapeutic strategies that boost tolerance. So even in the absence of innate resistance or in the absence of interferon, mitigating some of the consequences of caspase 111 activation can give the host a chance to repair any tissue damage and clear invaded pathogens. So to conclude, I've shown you that MX1 mice serve as a great model for flu infection, which have been able to reveal the role of both TLR7 and RIG-I in immune resistance, which is something we couldn't do in red mice before. And we've shown that in the absence of TLR7 and RIG-I, there is increased viral titer, but there's also increased bacterial infection of the lung. And our data show that signaling downstream of caspase 111 and that neutrophil mitosis trigger inflammation and damage to the death. And this has important clinical implications for treating flu infection, possibly in the elderly, because Ryan showed us that those innate cells from old donors have impaired interferon and impaired responses, but intact caspase 111 signaling. So one potential strategy could be to use Palmazyme to stop some of that mitosis and give the host a chance to repair and tissue damage. And also, inhibition of caspase 111 itself could boost tolerance and improve survival after flu infection. So that I can think of a million more experiments I'd like to do with the product. But to name a few immediate for future directions, we need to determine the role of caspase 111 in mitosis. We have some data showing this already with my friend Tim, but specifically, is it due to neutrophil intrinsic caspase 111 signaling triggering mitosis, or is caspase 111 signaling happening in some other cell type that's triggering mitosis? Basically, in the neutrophil itself, is caspase 111 playing a role in mitosis, or is it coming from somewhere else? I'd also like to investigate if pyrotosis is involved. So pyrotosis is a form of inflammatory cell death. And pyrotosis occurs downstream of caspase 111, which is demonstrated in activators of the cell type. But I'd like to investigate if pyrotosis in neutrophils is involved in mitosis. Also, since our tolerant mice are caspase 1 and 11 knockouts, we don't really know whether our phenotype is due to caspase 1 or caspase 11. So we need to figure that out. And lastly, we have a drug, VS765, that specifically targets caspase 1. So I'd like to see whether we can induce tolerance in vivo by pharmacological inhibition of caspase 1 in those double knockout mice. So that actually brings me to the end of my talk. I'd like to quickly acknowledge people who have played a role in this project. And then we're going to take any questions around this work. And then after that, I'd like to probably thank everyone. So a quick thank you to my guy and Joe, who is and firing something. I specifically like to thank Ping, our technician up Ping, noise and innovators. I also like to thank our lab manager, Melissa, who is just absolutely incredible, and my friend and lab mate, Ryan, who's doing all that cool stuff with human. And I'd like to thank all our collaborators and my community members, Sue, Jordan, and Rosan, for all the support. And thanks for your attention. I'm going to take questions now. My brother.
So, there's two ways to state this guy. The way I see it, one is to, when you have aberrant or impeded innate immune signaling, you can do what you're trying to do, which is to attenuate the downstream cascade signal, right? To minimize tissue damage. The other thing you can do, based on both your data and Ryan's data, is you can do something to back up interferon signaling in individuals that are deficient at baseline, right? Now, the problem with that strategy is that we don't have the luxury of knowing the people exactly.
By the time they show up.
So let's say we lived in on this universe and there's an alarm going off when somebody got What do you think would happen if you flashed somebody with exogenous interferon alpha like one day?
Yeah, so basically, so I mean, obviously the kinetics of that also have to do with the virus and how awesomely the virus will work. Right? So if you have a really virulent virus, chances are if you turn on interferon and you turn on IgG, you might, in the initial stages of infection, be able to limit viral replication, right? But Ryan will be able to tell you someday, but basically we need to figure out in the elderly if there's some sort of defect in the amplification of the interferon signal itself, right? So maybe if it's not just interferon that's responsible, and the amplification of that interferon signal perhaps after day 3 or day 4, right? You still need that interferon response in vivo. So I mean, so it's kind of hard to see whether it depends on how quickly the virus will replicate, and it depends on exactly what the defect is in the So
You need propagation of the signal.
you need propagation of the signal. So, but it could work.
So what with the, in terms of actual IST induction, you take the model that you take cells from the other people that are done so that post IST induction, right? So it's not a default.
Sometimes expresses MS1 in the brain. In that the original table, they give a cranial injection of fluid. And where I'm going with that is there a link between MS1 expression in the brain and rare cases where fluid causes encephalitis in human cases?
Yeah, so I mean, I don't really know much about fluid infection in the brain, but MS1 is pretty much ubiquitously expressed in like almost every cell type. So in those cases, when flu does infect the brain, it's not necessarily used, lack of MS1 infection there necessarily, but MS1 is expressed in the brain.
So actually, one thing that
I'm deleting my names.
One thing you asked me about mice, I think there's so much redundancy in the immune system. It kind of almost surprises me that this is the one critical effector involved in regulating this pandemic.
Yeah.
So what? I mean, doesn't that surprise you?
Yes. Yeah. So I mean, so there's a lot of cool things there. So yeah, I mean, one thing also is it'd be interesting to see, there's so many other studies looking at the role of other ISTs, but it's done in MS1 conditioned mice, right? So there's a chance that, on the correct background, that maybe some other ISTs do have a more robust role, right? But coming from an extremely biased point of view, MS1 is like the coolest thing. So MXA in humans, it doesn't just restrict influenza, right? So different portions of that molecule have evolved to bind and restrict a bunch of other viruses, measles, hepatitis B, and all sorts of other viruses. So, but yeah, it's very interesting if you see such an extremely potent role of MS1 that's nice to say. I'm a tough family.
What do you think contributes from the figure that you showed of when the mice were treated with palmazine? What do you think contributes to the high degree of variability of body weight that we saw?
Yeah, so you mean in the mice that were treated with palmazine. So there's a lot of stuff going on there. So one is that definitely it can't be just mitosis that's triggering mortality, right? Like that's just a major contributing factor. But it's also that I was giving this drug intraperitoneally, right? And somehow it's actually, I'm surprised that you can see effect, right? It's getting to the lung to have that effect. So I think a lot of that variability has to do with how effectively the drug is getting to where it needs to get to. So that's one way of looking at it.
So that was like the question I wanted to ask you. Do you know that chromosomes actually dissolving this in the lung?
So what I did was I did an experiment by FACS to look at H3 staining after. So what I did was I just had one experiment where I tried to look at that, and I'm going to spend more money and actually do it by histology way soon. but just by FACS, if I treat the mice with palmazine and then take the lung out, and then look at H3 staining in the lung, I can see less H3 staining. So I do have evidence that it's working. Okay. All right. So I've been here for a while, and I have a lot of people that I'd like to thank, and I'm going to try to hold it together. And I'm going to do this really fast and stay here for a while. But there are so many people who I need to thank, and without them, there's no way I can do it. Okay, hold it together. So first and foremost, I'd like to thank my mentor, who's been has been so incredibly supportive. Her scientific rigor, her creativity, and her excitement is so contagious. And because of her, after all this time, I still feel like I'm not doing things. It's just incredible. And she's talking so much about being a scientist. She's talking so much about life in general. And she's just so much fun to hang out with also. And it's just been such a privilege and honor to witness your scientific prowess and be in your lab. And I look forward to your mentorship throughout the rest of my career. And she's also so supportive of like all my other craft, all the music and everything competition, which is just incredible because she's so busy, she really does not need care. I also like to thank my powerhouse Thesis committee. Ruslan is suspended for 2 weeks, but it's been a privilege to be guiding Ruslan during grad school. He obviously, as you all know, has a totally unique way of looking at the problem, and I'm always excited to hear his new ideas about my project and about science in general. And I really want to thank Dr. Jordan Fuller. So when I wanted Jordan to be on my committee second year, he kind of asked me, it wasn't directly relevant at the time, and I'm like, I don't need a committee. And I explained that whenever we work at RIT in our research and progress talks on Friday, he always has such rigorous scientific feedback, and I always appreciated his questions for other students. And whenever I email Jordan, he always responds to all my questions immediately, and he has amazing feedback and insight, and his knowledge encyclopedia, his expertise has been tremendously important for my project, so I want to thank him. And I also want to thank Sue. Mom can tell you that I was already a huge fan of Sue before I came to Dell. And when I was deciding where to go to grad school, Sue and I had so many phone conversations about how I would make this decision because it's not just me, I'm from here first time, and she's so helpful in both of us moving here. And then when I got here, I got to see her creativity and excitement as a scientist, which is so important. It's a really long process. But whenever I'm in her office, so excited, and I have all these cool ideas, some of which I'd love to do, and she's also super fun and hilarious, so I really want to, okay, I'm going to pass it, okay. So, as Bill stated really perfectly in his defense last week, people has curated this group of amazing people to speak in her lab, and it's really a trickle-down effect from her, but it makes for such an incredibly supportive and productive place. I specifically want to make a shout-out to my cool crew, Leonardo, MJ, and Maury, and my baby Ryan, who's right over there, Paul next to Will, and he's just an incredibly talented scientist, and he's this awesome guy with it. And if you look over here, this is Hannah. Hannah's over there, and I rotated with Hannah when before I joined the lab, and she's just a brilliant scientist, and I love her so much. And she's now a professor at Washington in St. Louis. And I also like to thank MJ, and you can just tell by her smile right here that MJ has this infectious, very powerful smile, and I really need her positivity all the time. And she's taught me so much in the lab. I really appreciate that so much. And then there's these two people here in the middle, Will and Maria. So, Maria's always right here. Maria is my baby, she's my friend, she's my life advisor. She's one of those people, Maria and her husband Will, those people like, when you meet them, and you're like, I want our children to hang out someday. You know what I mean? That's the way I feel about Maria. It's just so important, and Will, so okay, Will and I met when we interviewed at Yale, and we were sitting at this table, and Barbara, she didn't know whether I was a vegetarian or a vegan. I'm a vegetarian, but Will, so she got me a vegetarian meal and a vegan meal, and the vegan thing was this beautiful platter of hummus and veggies. It was pretty big, so I put that kind of like next to me. And I was sitting there talking to the other recruits, and some punk starts eating my food. I was like, you're eating my food, but it was Will. I was like, oh, sorry. Anyway, and then we both came to Yale, and we both did I mean, we've been through everything together, and I just adore you so much. And just like you said last week, I have no idea words, but you mean so much to me. To the entire immunology department, we start over here with Lori, who's does the full meeting. He's so awesome. And there's Lori in my lab, and Anthony, who's also over there. Anthony is now a professor at Fred Hutch in Seattle, and this is the entire happy hour crew over here. A bunch of drunks. I love you so much. And there's Barb, our department admin, who's just so amazing and done so much for all of us. And my good friend Helen, who is now in California at Berkeley, and this is a student in our lab. I had her, and there's so many other amazing people in the department. Okay, then there's Sarah and Dan, and Sarah has a little bit of a grown-up now, but in her set. But he was one of my first friends when I came to Yale, and definitely one of my first friends as well. And both of you guys mean so much to me. I love you so much. And then Jeremy, who you might know by a different name, over there. Jeremy is like really smart. So Jeremy's already, he started the same lab. He's already a professor at and we actually became best friends because we realized we both love super so much. And even though he's so far away, you and Helen just mean so much to me. And I hope you do. And Megan's not even, Megan's friend and family. Megan's Christopher's cousin. And it's one of the reasons he came to Yale. Megan was already here doing grad school. And he's so special to us. And I'm so glad he's here today. And Lauren could be here today. Lauren, who I'm sure many of you know her, who's just been so wonderful to us. Our friends Tom and Rachel, who are just such an amazing, wonderful, warm, affectionate people. This is Rachel of Rock Island. And first time I officiated their wedding. And Derek is CEO of First Company. He's just in addition to being amazing for First supportive of First he's so supportive of me. And he's here today and it means so much to me. And both you and Nikki just rock my world. And I had and this is Dwayne. Dwayne is here too. And Dwayne means so much to me. You know, we've been in grad school for like, what, 5 years, 6 years, which is like the length of elementary school. And Dwayne has bringing me to school almost every day for the last 5 years. And it's not just like, oh, I'm going to do my driver to get on. You know, like every day he gets on and he's so sweet to me. And he asks me about my manuscripts and my experiments. And he's here today. And it means so much to me. And all my loans here, including Smith, who's also a postdoc in our lab, who's just brilliant and amazing. I just want to bump it right up. This is very their weddings, and you guys are amazing. And Ryan Hayes is here as well, so we adore you so much. And Officer Kriebel and Noel. These are like the first faces I see in the morning and the last face I see at night. I adore you guys so much. And Will and Shower getting married soon. Good Yale citation. This is my acapella group. And you know, one of the coolest things about Yale, and one of the reasons why I came here, is it's not just, oh, you're going to be in your lab for like 4 years and you're not learn to talk to anybody or scientists. There's so many opportunities at Yale to hang out with people from all over the place. And so this is my acapella group, and there are people here from law school, people from the medical school, people from the School of Forestry, and all of us that hang out together and sing together. And actually, you can recognize this photo, which is the one we use for defense. I'm actually in the bosom. But I'm going to miss seeing you guys so much. And Andrea, you just been such an amazing friend. She's the musical director of the group for so many years. And I just adore you so much. Okay. Then I have to thank the sound. So like, what are the chances that I show up at Yale and sing in the stands with like these really brilliant professors that are world famous? I don't know why I'm asking me to do this, but it's like a great thing ever. Richard's not here, he's in California, but it doesn't matter because his better half and I'm here. And Len, where are you? Oh, Len. So Len is actually, I'm going to blow up his spot right now. Len is actually dishing the talk of the Nobel Prize winner right now to hang out with me. And that means so much to me. And just to give you an idea, Richard opened up with animals back in the day, right? And this amazing scientist. Len opened up for Eric Clapton at Royal Albert Hall. He was but he's also a professor of pharmacology and cellular physiology. So these people have taught me that you don't really have a choice. You have to do music and science at the same time for everything. But it's been such an honor. And Bill's here with Katarina as well, our drummer. And you guys have just been so sweet and some like child who wants to sing with you. And it means so much to me. And specifically, I want to thank Richard and Maddie because they've just been so kind and they've treated me so well. Maddie treated me like a daughter, and I have no idea why. And you just and you play violin and cello together, and I love you so much. And I'll be eternally grateful, and I'm not leaving it. No matter where. And then I want to tell you about some awesome things. Okay, so I was diagnosed with this really weird mutation a few years ago, and I lost like 4 months of my PhD. But it doesn't matter because I was just constantly surrounded by amazing friends and family who kept me going. I started living in college in California, who I think is also here. And there's not much known about this disease, but whenever it is, it makes it all better. And Memorial's there all the time, and Helen's there all the time, and very less too, but I don't know. And Maria brought me food every day. Ellen, who came all the time, all my friends. Sarah came all the way from university to bring me to She was there all the time. The entire gypsy staff, when I was released from the brigade, the gypsy staff, organized by Lord Luce, had a dozen cupcakes from T-Gas Cupcakes and Cambridge delivered to my parents' house. It's delicious. But anyway, it was really tough time. It was really scary, and this was thankful. And then there are these people from Boston who are here today. I'm going to start with Krishna over here. And Krishna and his brother Mohan are my childhood friends. And our parents have been friends since the 70s. And I love you so much. And you know that I got your back, and you got my back forever. And I'm so thrilled that you're here. I believe that so many of you have met him. So, this project has to do with viruses, and all of a sudden I see the phenotype in neutrophils. Officer people, I already gave you your shout out, Rob. And then all of a sudden I need to work on neutrophils. And my best friend, the whole thesis on neutrophil neuroses. And so now she gets to hang out and we get to do experiments together. But she means so much to me, and I love you so much, and I can't wait for your scientific career to just work. And you're just brilliant. Then some of you know Sheldon. I've known Sheldon since I was 15 years old. So if you went to Harvard, Sheldon is the director of the Engineering Department. And he also taught high school chemistry at my high school. And he is responsible for any of the music that I've done since I was 15 years old. We performed for Desmond Tutu, we performed for Bobby McFerrin, we performed for everyone. It's all because of him. And most people by the time you're my age, you don't do music anymore. You kind of let that go. But because of Sheldon, I've never let go of it. And also when I was a rebellious teenager and I would listen to what anybody said, I would listen to what they said. So my parents, thank you for keeping me in mind. And lastly, I want to thank my best friend, Jen. Jen and I met on a 45-minute Delta shuttle flight from the Florida Airport in 2007. And we've been best friends since. She knows me better. Anybody? I don't think you guys do much. But I love you so much. And I'm so happy that you're here, and now you know why. I don't call you back here now. I love you so much. I can't wait to come home and hang out with you. And then my in-laws are on site, or whatever you call that YouTube thing. They treat me like their own daughter. And I'm just so thankful for everything they've done for me, how much they care from 3,000 miles away. It's just been so wonderful. And my my lunch, like the most selfless woman you've ever met. And I adore her so much. I really do. My Uncle Mario was his man, but flown in all the way from Minnesota for this. And we've always taken such good care of my mom and my uncle, they're close, so close. And they've taken such great care of both me and that. And I'm so happy they're here. We visit us each year. Well, not Suzanne visits us each year, but I think Uncle Mario can come to the park and stuff. But that's okay because we're going there. I'd also like to thank my cousin, who's basically our brother today. And he's just such a sweet guy, and I just adore him so much. And we've always been so close. And I'm so glad that you're here today, and I'm loving someone. Oh God. And then there's Romeo and Alex. So Romeo is actually Krishna's sister, but she's basically my sister now. And then there's the problem with this is usually I don't really do the emotions of talking about this kind of crap, but I just have. And so usually what happens is that I say some stuff, and then Romeo goes, well, what? How many of you? This is not really good. You know, but I can't do that right now because I'm talking about you, so this is going to be really bad. But I just adore you both so much. You're such special people, and I love your souls. And to be honest with you, when I was in the hospital, they came and they took care of me, and they communicated with the doctors and did everything. I really would not be alive today if it wasn't for either of you. And you're both independently such special people, and then you're also together, and it's to this like powerhouse amazingness. And then my dad and my mom. So there's one person more excited about today than me, and that's my dad. And Dad's also a scientist, he's a chiropractor, he's a pathologist, and every day he calls me and says what I'm doing and what experiments I'm doing. I think Akiko is kind of like tired dad. He call me at like 8:30 and be like, are you up? And he'll call me at 10 o'clock and be like, oh, what are you doing? Did you hear back from the journal? Like, every day. Anyway, I mean, I don't know where to begin. I'm just so thankful for how much you care about me. And I'm just so proud of my super liberal octogenarian feminist dad who only wants the best for me, who wants me to like, kick ass at everything scientifically and otherwise. And then there's my mom. And Mom is also super political girl. She's a gourmet cook. And I'm son by son where she's responsibility, strength, and unconditional love for us. Mom has kicked cancers left 3 times since I started school here. And she's taught us all about our South-Indian roots, and about Western culture, so we can equally appreciate Indian music, and equally appreciate Led Zeppelin, and Saturday Night Live, and Simpsons. And she's just my confidant, my role model, my life coach, my music teacher, and my dreams to be a mother of peace. Okay, if you could talk to me for more than 10 minutes, and then I spent 5 of those minutes talking about my big brother. My brother's here all the way from California, and I've always, as you can tell, I've just wanted to be this way in my whole life. And unfortunately, he sets the bar so damn high. But he encourages me whatever I do. I'm so proud of him. And I don't know where to put on for that comment. But and then after, and he also married the most amazing person on love. Who's also my sister, and I love her so much. And she and my super space niece, Zadara, are in my computer too. And I love them so much. She's not even 3, she already knows like all the planets and everything. I love you guys so much, and I'm so excited that I got another sister who loves our family and loves us so much, and I love you so much, and I can't wait to watch out and just keep playing. So there's no way I'd be here without you today. So Krishna was originally the coffee shop guy. He was just a really hot guy who used to be in a coffee shop behind something hot. And somehow, and I used to hear him talk, he was really smart. And somehow we met and then we got married and then we moved here. And there's this saying, if you find someone who loves you the way Kanye loves Kanye. Without being pathological, we just found each other. And you get like every Simpsons, South Park, Zoolander, Super Troopers, Anchorman, and Will Ferrell reference. Someone's faster than I do. You grew up with my germophobia and my deep weakness and my penchant for old people TV shows like Mom and Family, which by the way is a timeless political satire. But you've established this amazing career here, and you're such a wonderful. If you've met Christopher, you know everything he says, everything he does is so genuine and straight from the heart. And you make me a better person, and you make me so proud, and I want to make you happy. And we established this amazing life here, and we made it to the scale, and we started this amazing journey together. We're all the adventures to come. Maybe we can also get it out of it. Okay, now.
Okay, so it's not actually over yet, unfortunately. She's going to have the committee meeting right now to deliberate what she's going to get at the issue. Assuming that she does, join us at the Toxic for Lobby between Champaign and Cake. Thank you.